We're going to start with how our skeletal muscles organized before we get down into the microscopic and submicroscopic parts of it. And so ultimately what we're going to get down to is all the way down to the muscle cell level, but we're not there. Do you see that there's multiple layers of organization before you get there? Okay, so let's go back up and let's start with a whole muscle. Okay, so a whole muscle, like let's say this is the long head of the biceps brachii or something, right? A whole muscle is going to undergo a lot of tensile strength when you contract it. So pretend that I'm super duper buff dude or buff woman, but if you put some testosterone in there, you'll get a little buffer. And I'm lifting up a freaking Volkswagen, okay? What is going to happen to my biceps brachii is there's going to be a whole bunch of tension generated. But the thing doesn't bust apart, right? So how do you maintain structural integrity despite a whole bunch of pressure? What happens is the skeletal muscle has multiple layers of organization of connective tissue around it that gives it structural stability. So what we will see is in skeletal muscle, if you use the prefix MYS or MYO, prefix or suffix, it means muscle. As you get smaller into the skeletal muscle, they start using the prefix sarco, which means microscopic muscle structures and refers to flesh. But notice that this entire skeletal muscle is going to be wrapped in a layer of connective tissue. And the layer of connective tissue that goes around an entire skeletal muscle is going to be, what kind of tissue would you use on the outside of a skeletal muscle tissue to give it structural integrity despite the fact that you are really straining on it? That's what I would use, and that's what it is. It's dense, irregular connective tissue wrapping all the way around the outside. It's really strong, okay? The layer that wraps all the way around the outside is called epimysium. What does that name mean? On or upon the muscle, which is exactly where it is, okay? So I've got a big layer of epimysium wrapped around the outside, okay? But then, to give it even more structural stability, what we will do is start going to the inside of the muscle. Do you see that the inside of the muscle is divided up into these little bundles? Do you see that? Okay. Each of these is going to be a fascicle. I've used that word with you before. What a fascicle is a bundle of skeletal muscle fibers. The reason you can tell the rectus abdominis is rectus is because if you can't see a fiber, you can see a bundle of them. That's a fascicle, okay? Each fascicle is also wrapped in connective tissue, and you can see it right here. Do you see this? Wrapped in connective tissue, okay? This is a fascicle. Let's look at the fascicle a little more closely, and we can see that the fascicle is wrapped in connective tissue called perimysium. Here's what the perimysium is, guys. It's just taking the same epimysium and making it slightly thinner, okay? It's like a thinner version of dense, irregular connective tissue, almost like a reeler, but with not very much elasticity in it. So it gets thinner and it wraps around each individual little bundle. Note to self. The perimysium is continuous with the epimysium is continuous with the perimysium. So you take this big thick sheet and then you make it thinner and wrap around the individual fascicles, right? So you're getting a really strong association here. So this is called perimysium, but then we're not done. We have another layer of connective tissue inside skeletal muscle, and it wraps around every single muscle cell or muscle fiber, okay? So do you see this layer here? Yeah? Take that and break it down further, and you will see around every single little muscle fiber, every muscle fiber or every cell, you will have a layer of connective tissue called endomysium, okay? And those three are all continuous with one another. Epimysium around the whole thing, then thin out and wrap around a bundle, and then you are perimysium, and then thin out a little further and wrap around every individual cell, and then you are endomysium. Now, can you understand now why when the dude lifts up the Volkswagen, his muscle doesn't bust apart? Because I've got continuous layers of connective tissue that are holding its structural stability. And what also rarely happens is that you almost never pull a muscle off a bone. I mean, it does happen every once in a while, but hardly ever. And why is that? The reason is because that epimysium that's around the outside is now continuous with the tendon. What kind of tissue is a tendon made of? Dense regular. So I have the dense irregular with bundles of collagen going under a whole bunch of different ways. I'm going to make it continuous with the dense regular, right? And then I'm going to make the tendon continuous with the outer layer of the periosteum. Outer layer of the periosteum is made of what kind of tissue, guys? Dense irregular. So what this does is it doesn't give me any seam. So if you think about fabric, where is this going to tear? At the seam, right? But what if I don't have any seam? What if the fabric is continuous and I've got a continuous fabric around the outside of the muscle that is continuous with the fabric, the collagen fibers, right? In the tendon that is continuous with the periosteum, then can you see why it's pretty unlikely for you to actually yank it off, right? Does that make sense? So all of that tissue is woven into the other tissue so that you get a really strong association. Now, does it ever happen that somebody yanks a muscle off a bone? Yeah, it does, but less frequently than you would imagine, really. Even with people who are lifting really massively heavy things, usually when it happens is when somebody gets hit from an angle that they didn't expect to get hit from, especially like on the knee, you can rip something off. But most of the time it doesn't. And even if you do, a lot of times you just tear it slightly off. You don't even tear it completely off. Does that make sense to you guys? Okay, so what we have is the epimysium around the outside of the whole muscle, the perimysium around each fascicle, and then the endomysium around each cell. But let us remember that we are talking about a cell here. Now, I acknowledge this doesn't look a damn thing like the cells you learned in 1610. But we will figure out why today. I'm going to tell you that, remember, a muscle fiber is the same as a muscle cell, right? Those two terms are used interchangeably. This is a cell. It's weird looking for a reason. That means that just underneath this endomysium, you are going to run into a cell membrane, right? Right? So the endomysium is superficial to the cell membrane. The cell membrane is going to be similar to all other cell membranes, phospholipid bilayer and that kind of stuff. It's going to have a new name, though. We'll learn its name later. Okay, so we've got endo attached to the tendon and then the periosteum. All right, so now let's talk about getting blood and nerve supply to each skeletal muscle, and then we'll break it all the way down until we can talk about how contraction occurs. All right, so what has to happen is each skeletal muscle, somewhere along the fat part, the actual body or the belly of the muscle, usually you have one artery going to, one nerve innervating it, and one vein coming from somewhere along the belly. Now, there are exceptions to that. Sometimes you have more than one, but at least one. And so what's going to happen is that artery is going to have to split into smaller and smaller arterioles, right? And then smaller and smaller capillary beds until every single skeletal muscle fiber that you have has to have a capillary bed snuggling it, because this is really energetically expensive tissue, right? So it's going to need a really, really rich blood supply. So rich that, look at this cool picture. I'll show you a cool picture. They stained this so that you could see not just the skeletal muscle fibers, but the capillary bed around it. So skeletal muscle, part of the reason it is red is because every single skeletal muscle fiber has to have a capillary net all the way around it. So part of the reason your steak is red, and steak is cow muscle, cow skeletal muscle, yeah? Part of the reason your steak is red is because there's so much blood flow around each cell. It's not the only reason, but it's part of it. That make sense? Okay, so you have to have a really, really rich vascular blood supply to skeletal muscle. And then the other thing that is also true is that you also need, for every single skeletal muscle fiber to be individually innervated. And that may not be a word that you're super familiar with, but I bet you can figure out what it means. Every single skeletal muscle fiber must be individually innervated. Yeah, the nervous system has to communicate with every single skeletal muscle fiber. And this is important because it's different than cardiac, and it also has some implication for damage and paralysis. So I want you to understand this. You end up talking about this a hell of a lot more in phys, but let me show you. What I mean is these three skeletal muscle fibers, 1, 2, 3, that's 3 individual cells, okay? They are all innervated by ultimately a nerve, okay? And what I'm saying is if this one is ever damaged, this fiber is paralyzed. It will never do anything unless healing occurs. And healing in the nervous system is definitely not guaranteed, okay? So I want you to imagine that if, for instance, this muscle right here, this muscle, I don't know how many fibers it's got. Each one of these is a fiber. So it's got hundreds of fibers, right? Okay. If if I have nerve damage that damages innervation to a quarter of those fibers, I am going to have muscle weakness, yes? But if I have nerve damage that damages innervation to the entire muscle, then what's going to happen to the whole muscle? Paralysis, exactly. Yeah, does that make sense? So each individual nerve fiber has to be told what to do by the muscle fiber has to be told what to do by the nervous system. That's different than cardiac muscle, isn't it? How does cardiac muscle get around the need for individual stimulation by every single fiber? What does cardiac muscle have? So you don't have to tell every cardiac muscle cell what to do. So pretend that I am a nerve. She is the first cardiac muscle, and they are all cardiac muscles. Why do I only have to tell her what to do? Gap junctions, exactly. Yes, that makes sense? Skeletal muscle doesn't have any. And with skeletal muscle, if you do not speak to every single cell, then they will never do anything. That means that nerve damage in skeletal muscle is really, really massive. It can have really, really massive impacts. The metaphor that I always use for this is like my teenager. Okay, so I have 18-year-old, 17-year-old, and 12-year-old. My 18-year-old's boy. And I imagine him being skeletal muscle fibers, because if I tell him, or ask him, because we ask in our family, if I ask him to take a shower, but do not ask him to also wash his hair, use soap, yes, put on clean underwear after he's done, yes, none of those things are going to happen except for the very first thing that I mentioned, which was take a shower. That's it, right? So he's basically a skeletal muscle fiber, and if I do not individually innervate each of those activities, only the one that I specifically innervated will actually get done. Does that make sense? So he's basically skeletal muscle fibers, and he does not appreciate that metaphor. He's like, screw you, man. You didn't tell me to put on clean underwear. Who on earth needs to be told to put on clean underwear? If you have clean underwear, that's the kind you want to wear, right? I mean, I know we've all been in the position in which, well, maybe not all of us, in which we didn't have any, so we wore a swimsuit under our clothes. She's like, yes. She's like, yeah. It happens, man. They're basically just thick underwear anyway. So you have to individually innervate these. This ends up being really, really important when you talk about damage. The nervous system damage is definitely not guaranteed to heal. If it doesn't heal, then you will have partial paralysis that ends up becoming permanent. When we start talking about the nervous system, which is right after we do skin, we will talk about nervous system damage and nervous system healing, and you'll understand how to predict whether it's going to heal or not. Okay, so this picture right here is kind of a cool picture that shows you innervation. So these guys are all individually innervated, and so you can see that if you cut that axon, this one would be paralyzed. If you cut that axon, ultimately, if you damage a nerve far enough up, you can damage innervation to the whole muscle, right? Because what they do is they come down and they split off and go into individual fibers. Okay, so now what we are going to do is we are going to talk about microscopic and functional anatomy of skeletal muscle tissue. And this will probably get a little confusing at first. I do have an animation at the end. I'm going to draw pictures for you. We'll work on it together. And it's one of those things that you might actually have to draw yourself a couple times in order to get it, but you'll be all right. Okay, so what I want to do is I want to go back and look at an individual skeletal muscle fiber. And saying muscle fiber is the same as saying what? Muscle what? Cell. Yes, so it is a cell. Now, I acknowledge that all of that 1610 work that you did doesn't do you a whole hell of a lot of good when you look at this, because it doesn't look anything like the little fried egg that you saw in 1610. So why the hell does it look so weird? It looks so weird because it has all of the normal cellular function. Yes, it has at least one nucleus. It's got mitochondria and smooth and rough ER and cytoplasm and all that crap. But it also has a special function, which is contraction, which is related to all of this special stuff inside. Okay, so a couple of issues that we have when we're looking at skeletal muscle fibers. First of all, it looks different than everything else. And then second, somebody decided to rename almost every damn thing in it. And they did it to me too. You can't even call it a cell membrane anymore, or even a plasma membrane. You have to call it the sarcolemma. Everything got a sarco. They still call it the nucleus, so we're glad about that. But you don't even call it cytoplasm anymore. You don't call it almost anything, so they changed the damn name of everything. And I don't know why they did it, but they did it to me too. They did it long before I took it. And so why the hell do you have to learn it? Because that's how they refer to it in physiology and in the classes past this. Okay. Luckily, it also correlates to all other muscle tissue. So it works for cardiac muscle and it works for smooth muscle. So at least you get a little mileage out of it once you learn it. Okay, so let's start looking at the structures. Okay, each individual skeletal muscle fiber is multinucleated, and you guys remember that, don't you? Okay, so here's a nucleus, here's one, here's part of one, here's part of another one. You probably don't care, but I'll tell you how it got to be multinucleated, just so that it can make little sense. What happens is during embryonic development, you have all of these little uninucleated cells called myoblasts, and then during embryonic development, they fuse, and when they fuse, they become this long multinucleated cell. Is there any advantage to that? Yeah, there is, because if I damage this part, yeah, you still have nuclei down there that can do protein synthesis and all that kind of stuff, and or protein synthesis if they need to. So it's unlikely to kill an entire skeletal muscle fiber. You can kill part of it, but it's still got like a commander in each part. Okay, so they're multinucleated, and remember, of course, that they are striated. Okay, what we are going to get to today is understand why it's striated, but let's just confirm that you guys remember. Of course, you can see the striations really easily. Today, we're going to understand where they come from. And then, I'll get those, all those. I meant to get those before now anyway. So, as far as an individual skeletal muscle fiber, they're really, really long. They can be up to a foot long, or if you're a super duper tall person, way longer than that. But they're usually microscopic in width. So they're between 10 and 100 micrometers. Do you guys remember what a micrometer is? It's 10 to the negative third meters. So they're microscopic in width, but they're really long. So what we're going to do is we're going to break one down and look at the components of a skeletal muscle fiber. All right, so let's go inside a skeletal. Well, let's go on the outside first. Okay, so if we look at an individual skeletal muscle fiber, let's look at this figure. Okay, the cell membrane around a skeletal muscle fiber is just like any other cell membrane, really. It's basically a phospholipid bilayer with a bunch of proteins stuck in. They didn't draw in the proteins because they're not particularly relevant right now, but they renamed it. And so it's not called the cell membrane anymore, it's called the sarcolemma. But what it is the cell membrane of a skeletal muscle fiber. Okay, which one of these is true? Ready? A versus B. A, the sarcolemma is deep to the endomysium, or B, the sarcolemma is superficial to the endomysium. The sarcolemma is superficial to the endomysium. One more time, you ready? Ready? One more time. A, the sarcolemma is deep to the endomysium, or B, the sarcolemma is superficial to the endomysium. It's deep to, yes? The endomysium is superficial to that, right? So this is going to be underneath, right? Because this is the actual boundary of the cell, right? Okay, so here's the sarcolemma. And then inside the sarcolemma, of course, you are going to have intracellular fluid or cytoplasm, but they renamed it. Now they call it sarcoplasm. Notice that there isn't a whole hell of a lot of room for sarcoplasm, but it is in there, right? Wherever there's not organelles, there's sarcoplasm. Okay, so the sarcoplasm has a couple of things that are relevant to you. Again, we know it's multinucleated, so there's multiple nuclei in the sarcoplasm. Can you start to see from this picture why the nuclei tend to get pushed up against the edge? Why they're peripheral? Because it's busy in the middle, right? Okay, so multiple nuclei, and I want you guys to think about this for just a second. Typically, skeletal muscle fibers have far more mitochondria than other cells. And some of you guys immediately get that, and for those of you who don't, somebody tell me why that makes sense. Why would a skeletal muscle fiber have far more mitochondria than most? Yeah, because it needs boatloads of energy. So it has a whole bunch of mitochondria compared. So here's a bunch of mitochondria here, okay? And then there are two other things that are true about the sarcoplasm but do not show up in the picture but are functionally important. Ready? Okay. The other two are also related to this. The other one is that inside a skeletal muscle fiber, in the sarcoplasm, we store glucose as glycogen. Consider it the glucose piggy bank inside each individual skeletal muscle fiber. Okay? And then we store one more thing that we need for this process. We store oxygen inside the skeletal muscle fiber on a protein called myoglobin. And myoglobin is similar to hemoglobin. Hemoglobin binds to and carries what around in the bloodstream? Oxygen. Myoglobin binds to and stores small quantities of oxygen inside the skeletal muscle fiber. So, glycogen is stored what again? Glucose. Okay. Myoglobin stores what? Oxygen. What's the primary place that you produce ATP in the cell? Mitochondria. Do you see that what I have just done is made sure that at any moment I have a little bit of stored capacity to produce ATP? Why is that important? Because my skeletal muscle is my emergency response tissue, right? Whether I've eaten anything or I have really poor cardiovascular blood flow to my skeletal muscles, if a bus is coming at me, I need to get the hell out of the way, don't I? Right? So you leave it set up so that you can always get a second or two of skeletal muscle activity. Does that make sense? So you always leave a little bit stored in there so that you can do that. Does that make sense to you guys? Okay. That ends up being really important in phys too. Okay. So that's in our sarcoplasm. Now the only thing that we haven't addressed is the elephant in the room. What the hell are these big old Vienna sausage looking things in there, right? These big old things, okay? Those are big bundles of contractile proteins. And I'm going to give you a preview of things to come. Here's what they do. Big old bundle of contractile proteins, kind of like a coaxial cable. You know, a coaxial cable is a cable made of cables or a cable made of wires, right? That makes sense? So this is called a myofibril. The myofibril attaches to the cytoskeleton at the end of the cell, and then when stimulated by the nervous system, it will contract. Yes? And what is it going to do to the length of the cell? It's going to shorten it. Okay? So what we're going to do now is going to break down the myofibril and start to understand how it gives the cell striations and how it causes contraction. Good? Okay, so the myofibril is going to be the basis of both the striping and the contraction. But before we go any further, I need to make sure you follow me. You ready? Okay. Do you see that this myofibril right here, if we just looked at one, we can start to see how you actually get striations? Because do you see that it's lighter right here and then darker right here? Do you see that? And then lighter right here, right? And when they all line up, they end up making the whole cell striped. Do you see that? Okay, so before we go any further, please note that this jagged line to this jagged line is going to be what we're going to break down. You ready? Only we're going to look at it two-dimensionally so we don't get too confused by it. You ready? So follow that and look here. Do you see the jagged line to jagged line? This would be the light part. We'll reinforce that in just a second. This will be the dark part. Okay? Ready? Okay. So this structure that repeats along the length of a myofibril, this repeating pattern of the way the proteins are arranged, is called a sarcomere. And the metaphor that I use for sarcomere to myofibril is the sarcomeres are like boxcars and the myofibril is the train. Yeah? So you line them up one after the other like boxcars on a train. So what we're going to do now is we're going to build one sarcomere. Okay? So building a single sarcomere is going to go like this. Okay. So you guys are going to build it with me. This will be important for anatomy. It's also going to be important for phys. So get it as well as you can and ask questions if you get confused. Ready? Okay. So the edges are called Z-lines or Z-discs. Doesn't really matter which one you use. The entire structure from Z-line to Z-line is called a what again? Sarcomere. Yeah, it's a sarcomere. And we're going to build it. We're going to see how it causes striping in just a second. So I'm not going to build every single part of this because I'm just going to focus on the parts that are anatomically important. And then later on when you get to phys, you'll add in a little bit more detail. Okay? So I'm going to focus on these red ones and the orange one in the middle. Okay? So do you see that the red ones are attached to the Z-lines and the orange ones are not? You see that? Orange ones have a little springy thing, but they're not directly attached. Okay? So the red ones are going to attach to the Z-lines and they're going to go toward the middle, but most of the time they don't touch. The red ones are called actin, and they are the thinner of the two, and they are long and skinny, so we call them filaments. What are they? They're proteins. Where would they have been made inside the skeletal muscle fiber? What thing makes proteins inside a cell? Ribosomes, exactly. Yeah. Okay. The nucleus gives it the command. But so you weren't wrong. You're just like one step back. Okay. Ready? So that's the actin thin filaments. And then the other part that we're going to focus on is the part that kind of floats in the middle and doesn't directly attach to the Z line. And this is called the myosin thick filament. Okay? All right. Now, let's look at how this causes the striping first. Okay? If I use this protein staining technique, if there is thicker, more dense protein, it will stain darker. Yeah? And wherever there isn't thick, dense protein, it will be slightly lighter. So, tell me guys, when I look at this area here, from here to here, do you think that's going to be the darkest or the lightest portion? Yeah, it's going to be the darkest. So let's go down to here for just a second. This is using transmission electron microscopy so that you can see the staining. Do you see that right here is going to be the darkest part? Okay. This dark part right here, from here to here, we call the A band. I'll tell you why it's called that in just a second. It's the dark part. And then from the end of one A band to the beginning of the next, it's going to be lighter, yes? Because you don't have myosin, and myosin is the thickest, densest protein. Okay? And we call this the I band. Okay? Got it? So here you can see A band, I band. Yes? And then this would be the next A band. And it would repeat over and over and over again. I'll add in one more after I make sure that you understand how this correlates to this. How does this correlate to this? Where am I now? Which band? I band, A band, I band. Do you see that they run perpendicular to the long axis of the muscle, just like you learned striations did? Okay, so I just have to add in one more thing, and then we'll start talking about contraction. Go ahead.
Did you say the myosin, the thick one, is what makes it dark?
Yeah, it's a denser protein, and if you use protein staining, it's going to stain darker. Okay. That's what it does. That make sense? Yeah. Okay. All right. So the only other thing is that, draw this a little bit better. Do you see that there's this area in the middle, which is going to be dark, but not quite as dark as if you had both of them overlapping? Do you see that? Like right here. Right? That's going to be dark, but not as dark, because it has myosin, but it doesn't have actin and myosin. Okay? That little area in the middle of the A band right here is called the H zone. Okay. Now I'm going to teach you a little bit about how it contracts, and then I'm going to ask you what will happen to those bands and zones when it contracts. You ready? Okay. So I want to add just a little bit more detail, just a tiny bit, and then we'll talk about contraction. Okay, so adding just a tiny bit more detail, what I want to tell you about myosin is that myosin, you can see, has little projections sticking up off of the myosin. And you can see them there. Okay, these are called myosin heads. And then the actin, you can't see them, but I'm going to use a metaphor so that you can get a visual of it. The actin has binding sites for the myosin heads. I think of them kind of like handles. They don't look like handles, but I'm going to draw them like that. Okay? So do you see these little guys right here? And then I'll show you how contraction works. These are called actin, actin, preview of things to come. And this is way more complex than this, but it gives you an understanding to start with, okay? So how does muscle contraction work? This is how it works, guys. When the nervous system stimulates an individual's skeletal muscle, what's going to happen is a whole bunch of crap that seems like magic at this point, but it isn't, okay? And what is going to happen ultimately is the myosin heads are going to reach up and grab the actin active sites and pull. And what's going to happen to the distance between the Z lines when you do that? It's going to shorten, exactly. And if you contract enough of the boxcars, you can contract the whole damn train, right? Enough trains, you can contract the whole damn cell, right? You can pull the whole thing. Does that make sense to you guys? Okay, so to make sure that you understand this, I'm going to do two things. First, I'm going to say, if it works the way that I say it does, and the hands, the myosin heads, reach up and grab the actin active sites and pull, I wanted you to tell me which of the bands and zones will change length upon contraction. So your choices are A band, I band, and H zone. Think about which ones, could be one, could be more than one, will change length upon contraction. Which ones? H zone, I band. Do you think the A band changes length? Isn't the A band just kind of staying right there and pulling everything into it? Does that make sense to you guys? Okay, yeah, got it? So, when I took phys ages ago, we still did animal experiments in our phys lab, and they don't do them here. And one of the things that we discovered, well, we didn't discover it, but we experimented with it, is that contracted muscle and relaxed muscle looks different. Relaxed muscle looks striped, right? Contracted muscle, since everything pulls into the A band, it's really dark, yes? So one of the ways that we experimented with that was we took a frog. Hi, frog. Feel guilty, but never have forgotten it, have I? We pithed the frog so he couldn't feel anything. Then we stretched the gastrocnemius muscle across a slide, a light microscope, and then we zapped it with an electrode. And then what we saw is the muscle went from being striped to almost completely dark. Striped, almost completely dark. Striped, almost completely dark. So what you will actually see is this, guys. Look, so from here, here's a relaxed muscle, right? This one has two sarcomeres. So you see your A band in the middle, yeah? Okay, and your H zone. I band, and then another one, right? Okay, this is partially contracted. Look at the length of the H zone. Do you see it? Shortened. Look at the length of the I band. Shortened. Okay, this is maximally contracted. Completely gone, right? The H zone is completely gone, right? And you can't see the I band at all. Does that make sense? Okay, so this description of how this actually works is called the sliding filament theory. Because think about this. Is the actin changing length? Is the actual actin filament, is this changing length? No. Is this changing length? No. What are they doing? Sliding past one another. What are they? Protein filaments. Hence the sliding filament theory of muscle contraction. Does that make sense to you guys? So this is the sliding filament theory. Okay. So the basis of skeletal muscle contraction is in the same base, has the same basis as skeletal muscle striations, right? It's all based on this this organization. So upon stimulation by a neuron, you will eventually know that it's called a somatic motor neuron. If that doesn't mean anything to you now, it will later. Then the actin and the myosin filaments will slide past one another, and the Z lines will come closer together, and the H zone and the I band will narrow, right? And the A band doesn't shorten at all. And neither of the filaments actually change length. They just change position relative to one another. Good? Okay, so let me show you guys an animation. Just a short little one. It's like 2 minutes or something to show you how sarcomere shortening works. Ready?
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I band becomes very narrow.
Got it? Okay, questions? Anyone? No? Okay. I don't believe you, but okay, we'll move on anyway. So what I want to do is I want to take this and put it all back together. I want to talk about the structural organization of a muscle all the way from the gross anatomy down to the filaments, right? And then I want to talk about it all the way back up from the filaments all the way up to the gross anatomy. So what we're going to do is we're going to practice these relationships. You ready? We're going to do it out loud, and I'm going to give you some prompts. Okay, we're going to go from big to small, and then from small to back up, to big, back up. Okay, ready? So we're going to start here. An entire whole skeletal muscle is wrapped in connective tissue. That connective tissue is called what? Which mycium? Epimysium. It's epiperiendo. Okay, so epimysium is around the outside. Okay. And a whole skeletal muscle fiber is composed of multiple bundles. A whole skeletal muscle is composed of multiple bundles of muscle fibers. A bundle is called a what? Fascicle, yes. Okay. Each bundle or fascicle is wrapped in connective tissue called what? Perimysium, yes. Okay. And each individual bundle, each fascicle, is composed of multiple skeletal muscle cells, also called skeletal muscle what? Fibers. Good. Okay. Each skeletal muscle fiber is wrapped in connective tissue called what? Endomysium. Okay. Deep to the endomysium, you're going to find the muscle cell membrane called the what? Sarcolemma. Sarcolemma. Good. Inside the sarcolemma, along with all of its friends, are big cable-like bundles of contractile proteins. Yes? Those bundles are called what? Myofibrils. Yes? And the myofibril is composed primarily of two proteins. The thin protein in the myofibril is called a what? Actin. The thick protein is called what? Myosin. The arrangement of actin and myosin gives striations. What is that repeating arrangement of actin and myosin called? Sarcomeres. Yes? That's the repeating arrangement of actin and myosin. You ready? Okay. So now let's go all the way from the micro back to the macro. Okay. I am going to make a muscle contract, and in order to do that, I need primarily two contractile proteins. The thick one is called what? Myosin. The thin one is called what? Actin. Okay. The arrangement of actin and myosin that gives stripes and also is responsible for contraction is called a what? Sarcomeres. Okay. Stack up the sarcomeres one after another like boxcars on a train. It makes a big cable-like bundle of contractile proteins called a what? Myofibril. Okay. Wrap up the myofibrils with all of his friends, the organelles. Put it in a layer of cell membrane. Right. The cell membrane is called what? Sarcolemma, okay? And a whole skeletal muscle cell is also called the skeletal muscle fiber, okay? Take that skeletal muscle fiber and wrap it up with a whole big group of them, and you've got a bundle of skeletal muscle fibers. The bundle is called a what? What's the bundle of skeletal muscle fibers called? Fascicle. And a fascicle is wrapped in connective tissue called what? Perimysium, exactly. Take a whole bunch of fascicles, wrap them all up together, make a whole muscle, and then the whole muscle is wrapped in connective tissue called what? Epimysium. Epi, peri, endo. Right? By the way, we will run into the same pattern when we get to nerves. Nerves have epineurium, perineurium, endoneurium. It'll be the same pattern. Okay? So, do you think that this is something that usually sticks the first time somebody studies it? No, it won't. Okay? So what you're going to want to do is you're going to want to study it. I would probably study it before I sleep instead of waiting until later when I've got 20,000 things to study, right? You don't even have anything hard going on this week because you're just doing axial muscles in lab, right? It ain't that hard. Although you do have your appendicular quiz. So, okay. So we will stop there for today. We're going to finish cardiac muscle and smooth muscle, and then we will do skin next time. Okay?